Hello. My name is Jones, and today we are looking at the HIV life cycle. Before we go into this topic, there are some terminologies that we need to familiarize ourselves with because of the significance that they play in the multiplication of the HIV virus. So we have the CD4 receptor, co-receptor, and the T lymphocyte. When talking about CD4 receptor, we are looking at a protein that is present on the outside of infection-fighting white blood cells. CD4 receptors allow HIV to bind to and enter cells. Co-receptors. In addition to binding a CD4 receptor, HIV must also bind either a CCR5 or CXCR4 co-receptor protein to get into a cell. T lymphocyte. This is a type of white blood cell that detects and fights foreign invaders of the body. Let us now look at the steps that are involved in the HIV life cycle. So we have the binding and fusion as stage 1, though in other literatures you find that they will put binding alone and also fusion alone so that you can have 7 steps. But if we put these together, binding and fusion as one, we are going to have 6 steps. So we have binding and fusion, we have reverse transcription, we have integration, we have transcription, we have assembly, and we have budding. So when we talk of binding and fusion, we are looking at the cycle at which HIV begins its life cycle when it binds to CD4 receptors and one of the 2 co-receptors on the surface of the CD4 T lymphocytes. So binding, this is a process of attachment of the virus to the T lymphocytes that have CD4 receptors. The virus then fuses with the host cell. Now, when you're talking about fusion, we are looking at the virus releasing the RNA and its genetic material into the host cell. Reverse transcription, which is stage 2. An HIV enzyme called reverse transcriptase converts the single-stranded HIV RNA to double-stranded HIV DNA. In the reverse transcription, the enzyme that plays a critical role is the reverse transcriptase. And the main thing that is happening within this stage is the conversion of the single-stranded HIV RNA to double-stranded HIV DNA. Let us now look at stage 3 of the cycle, which is integration. The newly formed HIV DNA enters the host cell's nucleus, where an HIV enzyme called integrase hides the HIV DNA within the host cell's own DNA. The integrated HIV DNA is called a provirus. The provirus may remain inactive for several years, producing few or no new copies of HIV. So, within the third stage of the cycle of integration, we have the enzyme integrase playing a critical role. And all this is happening in the nucleus where the HIV converted DNA is now integrated from the word integration there. So we are saying it's integrated with the host DNA. Let us now look at a 4th stage, which is a transcription. When the host cell receives a signal to become active, the provirus uses a host enzyme called RNA polymerase to create copies of the HIV genomic material, as well as shorter strands of RNA may be made of the RNA called messenger RNA or mRNA. So the messenger RNA is used as a blueprint to make long chains of HIV proteins. So during the transcription stage, which is stage 4, we are looking at the cell receiving a signal to become active, and the main enzyme that is responsible for this is the RNA polymerase, because it helps to create copies of the HIV genomic material. Okay, and as well as the making of shorter strands of RNA called messenger RNA. So this is all happening in the transcription. So the messenger RNA is the one that will be now used as a blueprint to make chains of HIV proteins, or to make proteins that are required for HIV to assemble, or for HIV virus to be made. Let us now look at stage 5, which is assembly. An HIV enzyme called protease cuts the long chains of HIV proteins into smaller individual proteins. As the smaller HIV proteins come together with copies of HIV's RNA genetic material, a new virus is assembled. So in assembly, what the particles that are being assembled are HIV proteins and the HIV RNA in order to form a new virus. So in the assembly stage, we are looking at the enzyme protease playing a critical role in cutting long chains of HIV proteins into smaller individual proteins. And then we have the last stage, which is budding. The newly assembled virus pushes out, okay, so it pushes out like a seed coming out of the soil, buds, okay, so it buds out. So this is happening when the newly assembled virus buds out of the host cell, creating walls within the host cell, okay, and forming its envelope. So the newly assembled virus pushes out or buds from the host cell. During budding, the new virus steals part of the cell's outer envelope. This envelope, which acts as a covering, is studded with protein, or is coated with protein or sugar combination called HIV glycoproteins. These HIV glycoproteins are necessary for the virus to bind to CD4 receptors and the co-receptors. So the new copies of HIV can now move on to infect other cells. So this now leaves the cell that has CD4 receptors vulnerable, hence reducing the number of CD4 receptors. Let's have a summary of what we have just been looking at. So during binding and fusion, we are having the HIV, okay, receptors, that is CCR5 and CXCR4, and the CD4 receptors, where the glycoprotein will bind to the host cell as it infects it. Okay, so it will bind to that. Then, during the process of fusion, the HIV genetic material enters the host cell. Then, during reverse transcription, the enzyme reverse transcriptase is going to change the HIV single-stranded RNA to a double-stranded HIV DNA. Then when we talk of integration, we are looking at the enzyme integrase that is combining the new HIV DNA to the host DNA. Then during the process of transcription, we have the RNA polymerase that creates copies of proteins needed for the HIV. And then we have the messenger RNA that are used as blueprint to make long chains of HIV. In the assembly part, we have the protease that cuts small proteins particles that are needed for the HIV to assemble or to come together. So we have the HIV RNA and the assembled proteins to form the new virus. When these assemble together, they come out of the host cell. As they come out, they acquire an envelope. Okay, so an envelope of glycoproteins that is going to have the GP41 and GP120. Okay, that is needed for the virus to infect other cells. So that is what we have been looking at today, which is the HIV life cycle, and that is what is involved during this cycle. Thank you very much, and keep studying.